The key space we're working in is really trying to change the care paradigm for amputees. The primary problem is that right now the stump and socket model is very painful for the amputee and quite costly for the healthcare provider. So what we're doing is we're creating an implant that allows the loads of the prosthetic to be transferred directly into the skeleton. We think of our device as the USB port for prosthetics, a standardized connector allowing every device to connect in the same way. So right now we have prototypes and manufacturing blanks. We're going to be moving into our first animal model within the next 3 months, and then we'll be moving into first in human by start of 2018. The biggest advantage of the OneStart award for us would be being able to pay for our pilot proof of concept trials in animals and really get some more solid data behind the technology. It will mean that for amputees, they can get the high functionality devices, which are really restricted to less than 1% of the amputee population, and do so in a way where the healthcare provider will actually have to pay 60% less cost to care for that amputee over their lifetime.
Cancer is a patient individual disease that requires a personalized treatment, and in T-cell immunotherapy, a patient's own T-cells are armed with an antigen-specific receptor called TCR, enabling them to find and kill the tumor cells. The bottleneck is now the availability of TCRs for most of the patients. We at Captain T-Cell have developed an innovative technology platform that generates TCRs for all patients. These TCRs are highly effective because they target the cancer cells and they are specific for the individual patient. As proof of principle, we have generated a TCR that is specific for cervical cancer, and we are now in preclinical phase testing if this TCR is suitable for the clinic. Our product will be a TCR gene sequence that can be provided as digital information, and we will out-license this to a T-cell manufacturing company. With this TCR isolation platform, we in the end can provide TCRs that are applicable for a wider range of patients.
In the 5 years to 2018, $290 billion worth of pharmaceutical patents will expire. Now, to refill these lost revenues, they're going to need to discover new medicines. So we have a technology which enables that. So we have a novel platform which allows us to isolate very high quality antibodies, which are a type of medicine which binds to disease-causing proteins. And we can do that very quickly and efficiently. We have a very unique technology which allows us to specifically target antibodies to the business end of these proteins and thus have functional activity against disease. We tested our platform against 10 different GPCR targets and showed that we were successful against all 10. Now that's an unprecedented level of success. So we're now looking to raise £500,000 to discover a single lead molecule against a disease or a target that's involved in colon cancer. This could have a huge impact on the pharmaceutical industry in itself, allowing rapid and high quality discovery of new medicines. And ultimately what that's going to mean is faster medicines getting to market, cheaper medicines getting to market, and medicines against diseases which we currently have no treatments for.
T cells normally kill cancer cells as part of their natural protective immune response in the body. And so people have realized that they can use these cells by injecting them into patients to kill tumors. And so what we have developed is a method and a technology of actually generating these types of memory T cells for use in immuno-oncology. We are the only people that can do this currently. We have discovered a new pathway inside these cells that we can modulate to produce memory cells. We have a set of candidate molecules that we have shown to be working in vitro, and we have also proof of concept data in using in vivo mouse models as well. We have quite a lot of things to still do. Optimize and improve our lead candidates further, as well as incorporate our current technologies into the existing processes for manufacturing therapeutic T cells. Some of these T cell therapies are showing quite dramatic responses in the clinic. We're talking about cures here. We believe that our discovery and our technology can enhance these therapies further and drastically improve their therapeutic potential. and they harness the body's natural immune response against tumours. So it's a totally fundamental shift and paradigm-shifting way of treating cancer.
So each year, about 150 million new cases of upper limb impairment are recorded. These patients need physiotherapy, and at the moment this is incredibly resource heavy and high cost. So we've developed Grippable, which is a hand controller that records grip force and also whole arm movements. What it allows patients to do is to interact with games that we give to them, and the idea is to increase their motivation and to capture some of the assessments that they're performing. We're the only company at the moment that develops devices that interact with mobile tech that we can download onto patients' smartphones or tablets, so we can deliver the therapy straight into the home. We've done our first usability and feasibility studies. Almost 500 patients have now been using the device throughout our hospitals. The next stage for us is to develop the software side and to construct the company that we can be sort of self-sufficient, and the more therapy leads to faster recovery. So these patients aren't able to interact normally in their daily lives, so aren't able to perform simple tasks with their hands. And the idea is that if we can retrain this, it will really improve their lifestyle and get them back into a normal living.
21st century, we are still dying of cancer. Let's face it, the truth. So we want to try to create a new market segment with a new class of compounds. We are exploiting a new approach, a new strategy, being between chemotherapy and immunotherapy. Our peculiar ability to bioconjugate radioactive transition elements to biomolecules. And we exploit these biomolecules to target our final compounds to cancer cells. On the world, we are at the advanced preclinical level. We have studied both in vitro and in vivo our compounds, both in academia, but more recently we are working in the under GLP conditions, so good laboratory practice conditions. Next step will be to complete the advanced preclinical testing and to enter phase 1 clinical trials. It's a curve. Yeah, we have also some ideas for preventing cancer, to diagnose it at the early stage, but now our urgent stuff is to work on cures.
Antibodies are arguably the world's most important drug. For millions of people, they help to keep aggressive cancers at bay, suppress overactive immune systems, and neutralize pathogens. The antibody discovery process involves screening large libraries of antibodies for high performers. At LabGenius, we can manufacture the world's largest and most advanced antibody libraries. What sets LabGenius apart is that our technology enables us to produce the largest libraries with the most diversity and the highest fidelity. So we're shipping antibody libraries around the world now, and really we want to take our technology to the next level by developing complementary design tools that allow us to both design and build the world's best synthetic antibody libraries. Ultimately, being able to discover better antibodies means that we're going to be able to keep more cancers at bay, we're going to be able to treat more immunological disorders, and we're going to build treatments for dangerous pathogens faster.
Two % of newborns are conceived using in vitro fertilization, and more than 60,000 cycles per year are carried out in the UK alone. Nonetheless, IVF remains an inefficient process, with less than 1 in 5 transferred embryos actually implanting in the mother's womb. So the reproomics solution is to test embryo fluids, something like an early blood test. We employ a safe, minimally invasive and effective way to measure key protein biomarkers released by the embryo. And we do this both reducing the time the patients need to wait and the struggle they go through while pursuing their dream of starting a family. Our advantages over other molecular techniques employed today are a lower degree of invasiveness and an extremely easy implementation in the IVF lab. We published proof of concept of our technology, and we're now looking to employ it clinically. We need lab space, we need to develop software that is able to classify embryos' viability and to build a self-implementing database with the results.
We have developed a technology to do direct manipulation of the skin microbiome to basically address skin diseases like such as acne, rosacea and eczema. Nowadays, no cure or no quick and safe treatment. And what we do by manipulating the microbiome, we basically trying to solve the origin of the disease, not just the symptoms. We don't focus on one specific bacteria that is missing in us. We really think that only complex mixtures and balanced mixtures can make your skin healthy and balanced and stable for a long time. And we have filed a provisional patent on our technology. At the moment we are doing the first clinical testing in healthy individuals. But the next step is now the most important, is the clinical testing in acne patients. And I think at the end of the year we would like to raise kind of a seed round of further development. Our technology is kind of a platform technology, and with growing research we would like to target other skin diseases, as mentioned, as rosacea, eczema, maybe even aging, lifestyle. The use of live bacteria is huge, and we see huge potential in the future.
I founded the company as a medical student. I witnessed many patients deteriorate because the warning signs were recognised too late. I thought this was crazy, so left and founded the company to solve it. We put a wearable device on the patient's upper arm. We monitor them across a very wide range of indicators, transmit that data off, and then interpret the data in software to say this is the patient that the clinician or nurse needs to go and see. So we see ourselves in any environment where only 1 or 2 healthcare staff are trying to monitor a large number of patients. We use a single medical grade wearable device that monitors across everything the health services need. A lot of our competitors use multiple devices or don't monitor enough indicators. So we've built an outstanding team of 6 people. We're in our first product trial at the moment, and we've raised about £600,000 so far, about 80% of which is non-dilutive. It's not just about the money, it's about the contacts and reputation that winning the competition wins that help us as a business. We see this as a global business that could potentially be extremely significant.